Hey everybody, it's Mr. Schultz. We're going to get started on our next set of notes. We're still talking about cell division. We're going to go into a specialized type of cell division called meiosis. It's a little bit different than mitosis, so we're going to go right into this and get this going right away. And right at the end, we'll talk about the different assignments we got this week. All right, so we're still covering obviously cell division. Now we're moving into something called meiosis. Here are the learning objectives. You can find them also on Canvas. And then I also want to just touch on a few vocabulary words. This is also on Canvas. You'll need to know what the difference is between haploid and diploid, and we talked about that last week. You'll have to know what a gamete is. Basically, a gamete is a mature egg or sperm, reproductive cells, if you will. Somatic cells are anything other than reproductive cells. And then we're going to talk a little bit about crossing over. I kind of introduced it, just briefly mentioned it last week. And then meiosis, which is a type of cell division that results in gametes. Okay, so we'll jump right into this. And again, these vocabulary words are also on Canvas, so you can go back and review them. All right, so meiosis is a type of cellular division where the daughter cells are genetically different. Mitosis, they were identical, right? The parent cell gave rise to daughter cells that were basically clones of one another if everything's working correctly. This is extremely important to the idea of increasing genetic diversity. So this is done during sexual reproduction when we're forming these gametes, sperm and egg. So if you remember back to the beginning of the year, we learned about ecosystems and the idea that more species an ecosystem has inhabiting a specific area, the more stable it is because of species diversity. Well, the same is true among members of its population. The more diversity we have in a population, the more stability we have. So the more genetically diverse they are as a species, the better chance they have to survive during environmental changes. One of the ways that species like humans are able to achieve genetic diversity is through sexual reproduction. We're going to talk about that a little bit as we go through there and kind of specifically focus on meiosis. This is different from asexual reproduction like binary fission, which we also talked about last week, which is what bacteria and other single-celled organisms kind of do. And this is because the offspring end up with different sets of genetic material. You get 50% of your genes from one parent and 50% of your genes from the other parent. The way this is achieved is through the formation of sex cells or gametes that fuse to form a new individual. The process of creating gametes is called meiosis, which is a type of cell division. All right, so just kind of connecting this to what we talked about before, right? There's three types of cell division. We talked about mitosis. We talked about binary fission. We're now moving into meiosis. Mitosis and binary fission are closely related. In those two modes of cellular division, the daughter cells are genetically identical to the parent cell, as we mentioned. This means that the DNA in the daughter cell is the exact same sequence as the parent cell. One example of mitosis occurs in humans is the development of new skin cells. And we kind of mentioned this last week as well, right? If you get an injury, a skin knee cut, whatever, or just normal everyday loss of the dead keratinized skin on the outside of your body, we have to replace those, right? So these new cells are signaled to divide. Once the cells are separated by injury, the signal goes out and we start obviously forming a scab and all that stuff. But then we start developing new skin cells. We basically, our skin cells go through mitosis and create new skin cells to heal the wound. Therefore, it's important that these new cells have the same DNA. We don't want to have different DNA, right? We're not a different organism. We want those cells to all have the same genetic material. Binary fission is a mode of reproduction for prokaryotes, and we mentioned that before. This type of asexual reproduction ensures that all the offspring are genetically identical to the parents. So here's a few facts about both of these modes of cell division, right? Daughter cells are genetically identical to the parent. The parent cell divides once. Binary fission is a type of asexual reproduction. DNA is copied once during the replication process. Mitosis is important for the growth and development and repair of organisms. And then diploid cells are sometimes referred to as 2N, haploid 1N. The N stands for the number of chromosomes the organism is made up of. In humans, the N number is 23. right? So if we have a diploid cell, that means we have 46, okay, total chromosomes, 23 pairs or 46. So as we go through this, keep those few things we just went over, right? Those things about mitosis that we just mentioned, kind of keep them in the back of your mind because we're going to see that there are some differences with meiosis. All right, so let's kind of jump right into this. So here's meiosis, and this is, I'm just going to go over this briefly. There's, again, several videos that you can watch on Canvas, and you should watch on Canvas for some of your assignments that are going to really kind of walk you through all of these different steps. But basically, you're going to see some of the same or similar sounding names. When we talked about mitosis, we talked about prophase, metaphase, anaphase, and telophase, and then cytokinesis. Okay? Except the big difference with meiosis is we're going to go through two sets of these. And there's subtle differences within each of these phases, and it's really explained well in the videos on Canvas. But let's just quickly kind of, you know, preview what you'll see in the videos with these still images here. So you have prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. And that leads you right into prophase 2, metaphase 2, anaphase 2, and telophase 2 with cytokinesis again. So really you're kind of going through two divisions. There's two sets of these phases that kind of occur. And there's some differences that are going to happen. Replication only happens once, even though we go through two divisions. That's how we get from 46 chromosomes down to 23 chromosomes in an egg and sperm. And then once the egg and sperm come together, the 23 from the sperm and the 23 from the egg make another 46. So we go from 46 to 23, and then once fertilization occurs and we form a zygote, it goes back to 46. Okay, so, and you can see some of the interesting things that are going on here. A lot of this stuff will sound familiar with the centrosomes kind of separating and spindle fibers forming, and then the chromosomes lining up along the middle, and then they get separated during anaphase. And then you have the cleavage furrow forming, and we have the splitting of the cytoplasm. And then once we have those two new cells, we go through the whole process again, except there's no replication in here. Replication only happens once. So once we start dividing again, now we're separating the chromosomes, and this time we make 4 cells instead of just 2 cells. We have 4 cells at the end of this with half the number of chromosomes. All right. So there's two main purposes for meiosis. First thing is it creates new cells that are genetically different than the parent cell. Genetic diversity is vastly important in biology, right? The more genetic diversity we have, the more stable the population is going to be. And then two, to create cells with half the chromosomes as the diploid somatic cells that make up the majority of your body. So when they come together with another sperm or egg, it goes back to the full complement. So remember that somatic cells are the cells except sex cells or gametes that make up an organism. That means your skin cells, your liver cells, your muscle cells, your bone cells, your brain cells, your nerve cells, all of that, they are your somatic cells. They're diploid. Sperm in males and eggs in females are the only gametes in the body. So they have half the number of chromosomes. Making sure the DNA sequence contained in the sperm and the egg cell are different from every other cell in the body is one way that we can achieve genetic diversity or genetic variation. Another important achievement of meiosis is making sure that the number of chromosomes in the species stays consistent from one generation to the next. This is why meiosis has two divisions, so we can get the number of chromosomes from 46 down to 23 for the sperm and egg. So when they come together, it goes back to 46. And again, there's some great videos on Canvas, and you really need to watch those. That'll help you kind of go through these different phases and see these things in motion. All right. So what's the results of meiosis? What happens at the end of this? So at the end of both cell divisions of meiosis, right, we go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then back through prophase 2, metaphase 2, anaphase 2, telophase 2, and the two cytokinesises. And that brings us basically down to 4 cells that are all genetically different than from one another. This is due to the something called crossing over that occurs, and we're going to talk about that in a moment. With pieces of DNA that kind of overlap and switch spots with the homologous pairs during prophase 1. That adds to genetic diversity. Also, the chromosomes do something called independent assortment, and we're going to talk about that as well in the next couple slides. This results in a lot of possibilities. Different combinations, more than 8 million. Okay, so now you've heard, hey, the saying where people have looked at it and say, hey, you're 1 in a million. Well, you can actually correct them now and say, actually, I'm 1 in 70 trillion possible combinations of sperm and egg that could have happened, right? So 8.4 million possible egg combinations times 8.4 million possible sperm combinations makes approximately 70 trillion possible combinations that produced you. So unless you have an identical twin, okay, then you're 1 in a 70 trillion chance that those combinations came together to form you. So we also went from cells that were diploid to haploid. The importance of this cannot be understated. If meiosis didn't reduce the number of chromosomes during the process of cell division, we would end up with zygotes with double the genetic material. So you'd go 46 to 46, you'd have 92, okay? And I don't know what 92 chromosomes are, but it's not human, okay? Humans have 46 chromosomes. So the chromosomes would continue to double every generation, and this would be catastrophic, right? It basically would cause the cells to die. All right. So another problem that would exist without the reduction of chromosomes would be the number of chromosomes wouldn't be the same among partners, right? This would mean finding a suitable mate would be much harder because the number of chromosomes don't match. The zygotes wouldn't develop properly at all. It's essential for members of the same species to have the same number of chromosomes. So we got to go from 46 to 23, and that's vastly important. If that didn't happen, right, then we have different species, and the chromosomes wouldn't produce much viability. A good example of this is a mule. A mule is a perfect example. They're considered hybrids. They're not a species because they don't produce viable offspring. They're infertile. And basically, a mule is a cross between a horse and a donkey. All right, so that's just one example of what could happen here. All right, so let's talk about crossing over, as we mentioned earlier. There's two main ways genetic variation occurs in meiosis. They are crossing over and independent assortment. So we'll cover crossing over first, and we'll look at this picture. We'll kind of follow it along as we go through the next few steps, right? So what you have here is you have two chromosomes. One of them is kind of this bright pink color. Let me get this pointer going. And one of them is this kind of blue color. So you got the pink one over here, the blue one over here, okay? When these chromosomes during prophase, these are basically exact copies of one another. They're clones of one another, right? But when they line up, okay, when they line up during prophase 1, little bits of this information cross over. So the tip of this chromosome will swap spots with the tip of this chromosome, and we end up with 4 possible chromosomes or sister chromatids that can be divided into 4 cells. So we could have an all pink one. We could have a mostly pink one with a little bit of blue. We could have a mostly blue one with a little bit of pink. Or we could have an all blue chromatid. Any one of those combinations is going to lead to more genetic diversity. And remember, for humans, this is happening for 23 pairs of chromosomes. 46 chromosomes can do this. So if we take a look, and this is basically going to kind of outline what I just talked about, but we'll put it in words, right, instead of just looking at the picture. So in the picture, crossing over is illustrated. The process of DNA swapping is one way genetic variation happens during meiosis. So if you take a look, step 1, or the first one, is it says the pink chromosome and the blue chromosome are considered homologous chromosomes. This term means they code for the same traits, although they may be coding for different characteristics. For example, blood typing, you could be a carrier for the O blood type, but have blood type A on your other chromosome. So this means that they code for the same traits, even though they may code in different ways, right? The pink chromosome represents the chromosome this individual got from his mother, and the blue represents the one they got from their father. Wherever the chromosomes overlap and twist around each other, the DNA can be swapped from one chromosome to another. In this picture, we see how the tip of the blue one exchanges with the DNA segment of the tip of the pink chromosome, and we get 4 possible combinations, right? So this creates 4 recombinant chromosomes in the bottom row. Notice that all 4 have a different color pattern, and this illustrates that they have different DNA sequences. So that adds to the genetic variation. Although the picture only shows one segment being exchanged, in reality, a number of exchanges can happen more than once on each chromosome. So the more exchanges, the more variation that can occur with the original DNA sequence, and we end up with newly created sequences, which adds a lot of genetic diversity. We also have something called independent assortment, which adds to genetic diversity. So in this picture, right, we're going to see how chromosomes already have crossed over. The crossing over has occurred. So if you take a look again, you can see that you got like a little bit of red over here and a little bit of blue. So you got some variation here. But in addition to that, in addition to the crossing over, we also have these chromosomes independently assort. So let's talk about what that means. Um, this is basically what's behind independent assortment. The random order in which chromosomes line up to be separated during metaphase increases genetic diversity. So here's how this happens. So let's follow these steps. And I think there's one more on the next page. The first cell in the upper left of the graph shows the red chromosome pair on the left side of the equator. Okay, so it's over here. And obviously the blue chromosomes on the other side of the equator. This leads to new cells having both red chromosomes or blue chromosomes, right? The cell in the upper row on the right, the one red and one blue on each side of the equator. So you could have 2 of them over here, 2 blue, 2 red, or you could have 1 red over here, 1 red over here, 1 blue over here, 1 blue on the other side. Or it could be the opposite, right? Maybe the smaller blue is on this side and the smaller red's on this side and the larger red's on this side and the larger blue's on that. So you got lots of different combinations that can happen here. We're just dealing with 2 chromosomes here, okay? This leads to 2 new cells having 1 red, 1 blue chromosome in each of the cells. So if these are the possible combinations, you could end up with something like this. You could have both red chromosomes. You could have both blue chromosomes. You could have 1 red chromosome, 1 blue chromosome, or the opposite. The larger chromosome is blue and the smaller chromosome is red. So, and this kind of puts some traits with this. This could be brown eyes, black hair, blue eyes, red hair, brown eyes, red hair, blue eyes, black hair. So you get these different combinations of genes resulting from these basically different combinations of chromosomes. So the bottom row shows 2 different combinations of possible results when we started with the same chromosomes that were created during crossover. So this picture shows the genetic possibilities of an organism with just 2 chromosomes. For organisms like humans with 23 different pairs of chromosomes, the possibilities of different combinations increases greatly. The 2 processes of crossing over during prophase 1 and independent assortment during metaphase, both metaphase, allows for genetic diversity to happen in the formation of these gametes. All right, that kind of takes us through meiosis. Obviously, those form the gametes, and then later on, the gametes can fuse, egg and sperm, become a zygote, and then we go through a whole bunch of mitosis and differentiation, and we end up with a new organism. So just for the assignments, make sure you do both. Check for understandings this week. There's 2 of those little short quizzes, and remember, you can do those multiple times until you get 100%. And then you have the assignment 3.4, which is a mitosis-meiosis Venn diagram. Now, I'll kind of go over this a little bit on Friday for you guys. But I just want to let you know there's on Canvas, there's one link that you can click on and you can use it. I think it's free and you can draw and design your Venn diagram. The only problem is I tried this out and I tried to send it to myself and it doesn't really come through real clear, even when I'm emailing it. If you want to play around with it and see if you can get it to work, you could try. What might be a better option is, even if you use that program, is to take your cell phone and just kind of take a picture of it once it's done and then upload the picture and email that to me. Or you can do it the old-fashioned way. You can just draw a couple circles on a piece of paper and fill out the Venn diagram. I think most people are familiar with a Venn diagram. If not, make sure you come to office hours and see me, and I'll show you how to kind of do that. And then you have the Meiosis Science Notebook, and you guys have been doing pretty well on those. You watch the videos and then respond to the writing prompt that you have. And that's it, guys. That kind of takes us through the whole meiosis section. We have one more lesson to do for this unit, and we'll do that next week. And that kind of focuses on uncontrolled cell division, which talks about cancer and stuff like that. And then we'll be ready for our first big unit test. So hopefully you guys got a lot of good information on this. And remember, come and see me during office hours if you need any help. Have a good day.